Hello, good evening to all. At first, I welcome our honorable speaker and all participants. I would like to thank our principal madam, head of the department, and dear colleagues. I request Dr. Nabanita Kundu, our faculty member, to introduce the respected invited speaker who will enlighten us over his talk.
Thank you, Monisha Di. Good afternoon, everybody. Today, we are pleased to welcome Professor Abhijit Bandyopadhyay, a very special guest from Department of Polymer Science and Technology, University of Calcutta. Professor Bandyopadhyay is the Technical Director of South Asia Rubber and Polymer Park Circle, West Bengal. He is the former head of the department, former member of Senate and Syndicate, University of Calcutta. Our distinguished speaker is also elected Fellow of International Congress of Environmental Research, India, Managing Committee member of Indian Plastic Institute, Kolkata branch, Associate member of Indian Institute of Chemical Engineers, and editorial board member of 2 international journals. Professor Bandyopadhyay did his BSc, BTech, and MTech from University of Calcutta, and he did his PhD from IIT Kharagpur. As of this moment, he has been teaching in Department of Polymer Science and Technology, University of Calcutta for 16 wonderful years. Apart from this, he is a renowned research guide. Till date, 11 PhD students, 19 MTech students have completed their thesis under his supervision. He already published 99 research papers in reputed international journals. He has written 4 books and 2 chapters of books of different topics. He is also the owner of 2 patents. Professor Bandyopadhyay is the recipient of Young Scientist 2005 Award from Materials Research Society of India. He is also the recipient of Career Award of Young Teachers from All India Council for Technical Education, Government of India in 2010. Today, Professor Bandyopadhyay will share his expert opinion on polymer nanoparticles in drug delivery application. With this brief introduction, we welcome Professor Abhijit Bandyopadhyay. Sir, please.
Thank you, ma'am. I think I'm audible. I'm audible.
Yes.
Yes, sir.
Okay. Thank you, Dr. Mukhopadhyay, for this invitation, and Dr. Kundu for this introduction. Now, today is also a very special day for me that I have been invited to speak before this august audience. And means I did my BSc in chemistry, but after that I shifted to engineering part where we did a lot of applied chemistry, these things, your engineering, which is more of interdisciplinary kind, where we have learned this polymer, which is mostly a physical chemistry part and organic chemistry part, and then chemical engineering and all. And during my research, actually again I shifted to some biomedical work and some means where we can use some polymeric materials for diagnosis and therapy. So that way, actually, from chemistry, I shifted to this, area. So what I thought that let us give you some, some introductory ideas about drug delivery and the latest research that is going on in the area of drug delivery using polymeric nanoparticles. These are not yet commercialized, but very close to commercialization, this technique, and why this technique is so explored nowadays, or why this is a very, very important field nowadays, that I would show you in a very elementary way. I will not go into much detail, but only point wise I will show that why this is so significant nowadays. So let me share my slides. I have prepared a few slides for this. So I'm sharing those.
Sir,
Yes, sir.
visible.
So the topic of today's discussion, as already mentioned by Dr. Kundu, is that use of polymer nanoparticles in drug delivery application. And as I mentioned that I will be giving you in a very elementary way, very simple way. I will try to keep it in a simple, I mean, versions and very brief. So let us first start with what we mean by drug delivery. By definition, it is known as the appropriate way to administer the drugs through various routes in the body for the purpose of improving health. Right? Now, this field actually is highly interdisciplinary. As I mentioned, that you need the inputs from various branches, various fields, and of course it is not a very young field, means people are working in this field for long. Maybe introduction of the polymer is a newer part of it, but working in the area of drug delivery is not new. Now it has recently evolved to take into consideration these points regarding drug delivery. And what are those points? That drug physicochemical properties, body effects and interactions, improvement of drug effect, and patient comfort and well-being. And collectively, these are coming into the subject called controlled drug delivery. Controlled drug delivery. Now, controlled drug delivery means when the drug is being administered in a controlled fashion, right? In a controlled fashion, and the drug is released within the body at a controlled rate. Now, along with this, you can also hear another term. Maybe you have heard, that is what we call it as a sustained delivery, sustained drug delivery, right? And that sustained delivery is that when this drug delivery, that period is sustained, right? Means for a longer period, the drug is being released within the body, but that may not be in a controlled rate. So there is a difference between sustained drug delivery and controlled drug delivery, right? Sustained delivery means that when the drug is released slowly, and the release period is enhanced. Nowadays you can see many drugs, they are coming in that sustained your mode. Suppose this antibiotic doxed, which is frequently being prescribed for this COVID infection. I think you have seen that doxed SL, right? That drug is named as doxed SL. Now what is that? SL is that slow or sustained release, right? So that dox formulation is made in such a way that it is released in a sustained way, slow and sustained, but may not be in a controlled rate. Control rate means where the rate of the release is not changing, right? So that way it's a slight difference between the sustained and controlled drug delivery. Anyway, now you see that this drug delivery we can have either in the conventional route, where we can have enteral route, parenteral, enteral route, and others. Now enteral means that conventional one, that is through our mouth, this your stomach, then intestine. So this is the conventional route, what we call it as a enteral route. And parenteral route means which is not enteral. Suppose when we go for intravenous, right, when the drug is injected during emergency, that is a parenteral route, means which does not cover this route, that is your the stomach and your intestine and all. And other is the controlled delivery, where you have a sustained kind of delivery, as I mentioned, then extended period delivery, site specific delivery. Site specific, what we call it as a targeted delivery. Targeted delivery also nowadays means these once these polymeric materials are coming into this field for being applied in this drug delivery application. So we are also researching on the site specific delivery, that is the drug is to be delivered specifically to that organ. Suppose you have some liver problem, right? So the drug should specifically deliver to the liver. It should not go, I mean, other organs as well, which happens during the present therapy, right? In present therapy, what happens? A part only goes to the liver, and rest of the part goes to the other parts of the body, and that's why it creates so many side effects, right? So this site specific delivery, where the efficacy of the therapy you can enhance, so that way what we call it as a targeted delivery or site specific delivery. And pulsatile delivery means that is the in pulse mode, release will be in the pulse mode. I will show you one figure, there you will see that the pulsatile delivery. So these are the ways, I mean, conventional delivery, conventional route is with us, we know, and controlled delivery route, nowadays it is being explored, right? It is one example, again, I can give you, suppose patches. Nowadays we use drug patches. I think you have noticed in few cases we apply drug patches on our skin. Now that patch you can put it for 24 hours. So during that 24 hours there will be a gradual release of the drug through the transdermal route, which is unconventional and that is much controlled. So that is an example of controlled and unconventional route for delivering the drug. Now let us come to that first conventional route, that is oral administration. Now this route has several advantages and we prefer that. We prefer this route of, or this way of taking the drug. That is it is highly your convenient for the patients, it is not invasive, and it gives you higher compliance, patient compliance. And regarding the manufacture, it is a well established process, and it is with available infrastructure. Now it has a several disadvantage, right? Means unconscious patients cannot take the drug. Mostly what happens, the drugs are not all the drugs are not soluble in water, so that insolubility or low solubility causes a problem of absorption of the drug. Low permeability to the, I mean, your in the organ part also. Now sometimes these drugs taken through oral route are degraded by the gastrointestinal enzymes or flora, and it cannot pass the first pass metabolism. Now what is that first pass metabolism? Whenever a drug is taken, the activity of the drug while it reaches the organ is reduced severely, right? So what we call it, that effect is a first pass metabolism effect of the drug within the body. That is why what happens, that when we take drug orally, sometimes what happens, the therapy that we expect, we don't get that, and it is because of the higher extent of first pass metabolism. And that is why in emergency we avoid taking drug orally, rather it is being injected. It is injected so that the first pass metabolism, that part can be avoided and the efficacy of the drug is retained. Then the drug might interact with the food, or some irregular absorption problem can also be there. Now it is in oral administration, what we have tablets, capsules, soft gelatin capsules, suspensions, and elixirs, right? So these are the ways these can be delivered orally. Then you can have a buccal or sublingual, right? The advantages, when you go for this buccal or sublingual delivery, you can avoid that first pass metabolism, so the efficacy down of the drug molecules, that you can restrict that. It gives you a rapid absorption, and you can also have to face low enzymatic activity, right? Whereas the disadvantage is the discomfort during dissolution of the drug within the body, probability of swallowing, loss of effect, sometimes it can also happen, and only small doses of drug can be administered, right? So in this form, you can have chewing tablets or chewing gums, like these vitamin C tablets. It's a chewing, we used to chew it, right? And there are also some tablets that we chew it. So it is that way of delivering the drug. Then rectal route. Sometimes we go for the rectal route if necessary. So here also you can avoid that first pass metabolism effect. It is useful for children. Whereas disadvantage, the absorption depends on the disease state of the disease, the degradation by the bacterial flora that is present in the rectum, and the way of delivering is uncomfortable. So it is being delivered in the form of suppository or enema. So these are the forms which are available, which can be delivered through the rectal route. Then intravenous or IV. It has the advantages of that the drug is 100% bioavailable because it is directly injected. So we get the rapid response. So that is why it is delivered in emergency. The total control of blood concentration that is obtained, then it can maximize the incorporation of the degradable drugs, the drugs which degrade faster within the body. So if you go for intravenous route, that means the drugs is not getting enough time to degrade before the action starts, right? And it can also again avoid the first pass metabolism. The disadvantage again is the invasive. It is invasive way of delivering the drug. You need trained personnel, right? We means normal people cannot give you. Then there is a possibility of toxicity due to incorrect dosing. If the dosing is incorrect, then the toxicity can be a problem, and sterility of the drug. Sometimes this could be a problem. Now it is being delivered through injection or IV bags, right? That is infusion technique. Subcutaneous uh, subcutaneous technique, that is, it is advantageous because of the patient self administration. Suppose this patch we can ourselves can apply on our skin. It is a slow method of delivering the drug. The drug is released in a very slow way, so the drug gets enough time for complete absorption. And also in this route, the first pass metabolism effect can be avoided. However, it is a disadvantage because sometimes it could be invasive. Sometimes it may cause irritation, inflammation, and the dose administration is only 2 ml. You cannot go beyond that. So that is a restriction regarding the dose. Intramuscular, right, means sometimes we have that intramuscular injection. Suppose this your COVID vaccines, right? So these vaccines are being given intramuscularly, right? Within your muscle, it was injected. Now, it has the advantages that the patient can administer the drug himself, right? It can be administered by himself. Sometimes patient can do this. Larger volume than the subcutaneous means at least you can deliver larger volume. Suppose this 7 ml, right? In one dose of this COVID vaccine, 7 ml was injected. So it is more than 2 ml in subcutaneous route. And also in this way of deliberation can avoid this first pass metabolism. Disadvantage is again invasive. Sometimes it causes the patient discomfort, irritation, inflammation could be a problem, and it may require some training also. Means just as a layman you cannot apply. You have to learn it, but you can apply by yourself after learning. That is the way. Inhalers, we know that nowadays it is very common for the kids because of this your so much of pollution effect that we see. So in this inhaler, we have the advantages of again it can avoid these first pass metabolism, and it is being delivered in the form of that the pressure is released. So that is why there is a formation of the gases which are rapidly absorbed, right? Now, disadvantage that solids and liquids can be absorbed if size is below 0.5 micrometer. Means this particle size. If it is below that, then along with it, whatever solids, liquids are there, they also get absorbed along with it. So that is the disadvantage. Again, a transdermal, that is some kind of a patch, as I was mentioning. You see in the picture also. It has the advantages that it gives you a localized effect, right? Means where there is some local infection or something like that, it can give you a localized effect. And ease of administration means just here you don't require any training, you just apply the patch by yourself. But disadvantage is a low absorption for some drugs. If some drugs are particularly they are not water soluble, means insoluble, that is oil soluble, what we refer. So for those cases the absorption rate is low, and it may cause the allergic reaction. Sometimes it may cause. Now, in this case, the requirement is the dose is 10 milligram per ml, and the drug molecular weight should be less than 1000. Now, once the drug is being administered, then it is being excreted through this route, some through gastrointestinal route, some through circulatory route, and some through the metabolic site. So that way, the excess or unused drug is excreted from the body. And this is for the conventional therapy. Now, what are the factors that influences the selection of the delivery route for the drug? This is important, that how to deliver. You see, delivery route means whatever we mentioned, that conventional and unconventional. So collectively, these are the delivery routes which are available. Now, when you are developing some modern, say, polymer nanoparticle encapsulated drugs or simple drugs, right? Whatever be the drug you are using, either a modern form or the conventional form, you have to deliver through all these routes, right? These are the routes which are available, right? So that way, how to select the delivery route, that is very important, right? For the to achieve the therapeutic level and the therapeutic efficacy. So that way, how to select that, and these are the points based on which the delivery route is being selected. Say first is the drug physicochemical properties. So what is the molecular weight of the drug, right? What is the half life of the drug? All chemical substances, we know they have their half life, right? So that half life we need to calculate for the drugs also. If the half life is low, then the therapeutic time is reduced and you may not get that desired therapeutic efficiency from the drug molecule. So that way, half life, what is the half life? Whether it is, low half life or longer half life, that also is important to know. And let me tell you that using this polymeric part, your materials, these polymer drug conjugates and all, we can increase the half life of the drug. That is another advantage we can get by using polymeric material as an encapsulator, which increases the half-life. Say from, suppose if a drug has a half-life of 2 hours, you can increase it to 5 or 6 hours. So that is also possible with these polymeric materials. Now, chemical stability of the drug and loss of biological activity in aqueous solutions. So these are the points to be considered from the perspective of the drug. Then you can select the route. This is the first point regarding the drug. Then second is the solubility in aqueous solution of the drug, that is the hydrophobic hydrophilic balance, what we call it as a HLB of the drug, right? Suppose for the emulsifier we need to consider this HLB, right? So how much it is hydrophilic and how much it is hydrophobic, so that balance, depending on that balance, we have these parameters to consider before we go for the delivery route. That what is the pH, if it ionizes, what is the pKa of the drug, then the temperature, right, of at what temperature it would be administered, right, concentration, or also the temperature of storage of the drug molecules, concentration of the drug molecules, crystallinity of the drug molecules, its particle size and state of hydration. So these are the points that needs to be considered regarding solubility, particularly aqueous solubility. Then the drug biological interactions, right? That is when a drug is administered within the body, right? How the body reacts to it, right? And how the drug also interacts with the different, biochemicals that are present within the body. So first, whether it, how much it is sensitive to first pass metabolism, because this is the way by which the efficacy of the drug is reduced at a very first instant when it is being administered. So how much it is sensitive to FPM, that need to be considered. Low, if the drug has a low membrane permeability, right? That is the cell membranes and all, or the permeating the organs and all. How much it has that permeability, whether it is high or low. Then whether the drug is sensitive to enzymatic degradation or all, or any other enzyme like your biochemicals which are present, whether they are degrading the drug or not. Then whether it is also degraded by the bacteria or not. Suppose in the rectum or even in the our your stomach, the bacteria are present, right? So whether those bacteria will degrade the drug molecule or not, so that also you need to consider, again, the half life and the side effects, right? Then what is the desirable pharmacological effect, whether you need a local healing, local therapy, then you can go for topical or vaginal route of administration. Whether you need a systematic therapy, then you can choose for the oral, buccal, or intravenous kind of therapies. If you require an immediate response, that is, as I was mentioning, that is emergency. So whether it is, then you will have to go for intravenous, intramuscular, and nasal way to deliver the drug. Then also the dose size, how much dose is required. Suppose during fever, generally antibiotics are given, and if you follow that, if it is a 500 milligram, then it could be some two doses per day, that is two capsules per day. So per day it is 1000 milligram. Or if it is 625 or something like that, then it could be one. Now why that 500 milligram your that capsules are administered twice, whereas 625 for once? Maybe the requirement of the dose for the infection is suppose 200 or 250 milligram. But what happens when this 500 milligram drug is being administered, then it reaches that, or it your surpasses that therapeutic dose concentration, and it moves up in the blood plasma, that concentration of the drug. Then it follows very fast as well, it means follows down very fast as well. So that is why the therapeutic time is that the drug offers is less. So there is a rapid rise in concentration of the drug within the blood plasma, and very quickly it dissipates also. So that way the therapeutic time is less, and as the drug dissipates faster in general, the capsules, the mode, these gelatin capsules and all, I'm talking about that. So as it dissipates very fast, what happens that, say after your 7 to 8 hours, actually no therapy is available with the drug molecule, so as it has gone beyond the therapeutic level. So again you need another capsules to be administered, so that again it goes for that therapeutic level, and it gives you therapy for some time. So that way it is being prescribed for say two capsules a day if the fever is much higher, and if it is sustaining for say more number of days, then instead of two, some three capsules will be prescribed, because you need to sustain it, that drug sustenance in the body, that time requirement is now very critical, because maybe the infection, extent of infection is high. But when it is 625, in normal case it is administered once. Suppose that drug, I think, is known to you, clavam 625. So that drug is designed in such a way that it gives you some sort of a sustained therapy, and that way one dose is sufficient in place of two 500 milligram dose, right? But if it is a requirement of say three doses of 500 milligram, then two doses of 625 may be given. So it depends on that how much infection you have developed within the body. So that way the dose size determination is also important. And drug molecular size, what is the molecular size of the drug? Because sometimes we have a high molecular weight drug molecules, right? Even the polymeric molecules can act as a drug, and we call it in the I mean that forms a separate discipline, what we call it as a nanomedicine, where the polymeric molecule itself acts as a drug. So that way, what is the molecular size and the molecular weight? In generally, what happens that if the molecular weight is high, the molecular size also increases. So that way, not all delivery routes can give you good therapy or good administration of the drug if the molecular weight becomes higher. So that way, what is the molecular size and molecular weight of the drug, that also needs to be considered before determining the delivery route. Now actually, so this drug design and delivery, these collectively forms actually is a collectively is a it has two components, what we call it as a pharmacokinetics, and another one is the pharmacodynamics. So pharmacokinetics is related to the design of the drug, how much to be administered, how often, how long, and how it is to be administered. So that is a part of what we call it as a pharmacokinetics. And pharmacodynamics is the therapy that is we get out of this, right? Out of the pharmacokinetics, what is the therapeutic effect, how long it takes to cure, right? So that is a part of the pharmacodynamics, that is the effect. So here you see that the plasma concentration, in the y-axis and x-axis you have the time, therapeutic time, and you see the therapeutic window. So this is the drug concentration level to be maintained to get the therapy when you have developed some infection or any kind of problem within the body. Now you see that for the conventional one, that you see a faster rise and a very fast dissipation going below the therapeutic window to the no therapeutic effect that I was referring. Right, so you see the faster rise going above the therapeutic level to the toxicity level, because drug molecules are foreign materials. So when you cross the therapeutic level, it reaches to the toxicity level. Right, so once it reaches the toxicity level, it starts to dissipate faster the concentration. So you see the therapeutic time, how much time you are getting for the therapy, that is the time it stays within the therapeutic window, that time is very less, both in the when it goes up and when it comes down, right? Whereas the blue one, you see that where the delivery is sustained, means it is not going to the toxicity level, it is retained within that therapeutic window level, and it is continuing in that level for a longer period from the conventional one. So that is why, as this doxed SL I was mentioning, so doxed SL having this blue profile, because it gives you the sustained therapy. I mean, it maintains a concentration within the therapeutic window for longer period, right? But you see, it is not going in a constant rate. So it means that it is not a controlled delivery, but it is a sustained delivery, right? Had that been parallel to the time axis, so you can say that the concentration is not changing, so drug is released at a constant rate, so that what we referred as the controlled release. But here it is sustained release, only sustained release, right? And here you see that your this is the pulsating way of delivering the drug I was mentioning. And for this, the blue one is unsuccessful therapy, as it is going above, the your therapeutic window. And this one, this greenish one, that is a successful therapy, because it is being retained within the therapeutic your level or window. Now, this absorption of drug, when it is being administered within the body, it could vary within the body once this administration routes vary. Suppose you see that same 500 milligram of drug, when it is administered intramuscularly, it gives you a higher concentration, and it dissipates at a, it also dissipates faster, but it maintains a high concentration, right? Whereas when it is delivered orally, right, it gives you a lower drug concentration in the plasma, and also it dissipates but maintaining a lower concentration. So that is why intravenous is or intramuscular administration is preferred when there is an emergency. So the same drug, same patient, once it is administered orally, once it is administered intramuscularly, and you can see the effect. So that is why it is very important that which kind of administration for a particular problem has to be determined wisely by the doctor, that this should be the way to administer the drug, because it gives you a different effect within the body. Now, once we have this knowledge about drug administration and the problems that we can face with the conventional drugs which are available, then what was thought, that how these problems can be taken care, and that way these your concept of using polymeric nanoparticles actually came, or rather the use of polymeric nanoparticles in improving the drug efficacy that was being started to explore. Now, there could be four types of polymeric, nanoparticles rather, not exactly polymer, nanoparticles that are being researched nowadays in drug delivery application. One is the metal-based nanoparticles, another is the lipid-based nanoparticles, polymer-based nanoparticles, of course, and finally the biological nanoparticles, means made of proteins and all. Now, What's the speciality about the polymeric nanoparticles? We have several, advantages when we go for polymeric nanoparticles. First of all, they are solid colloidal particles ranging from 10 to 1000 nanometer. Now, it may dissolve or entrap or encapsulate or simply attach the drug molecules along with it. So, various ways of getting the drug polymer association, right? Because these systems have very high surface areas, these polymeric nanoparticles, drugs may also be absorbed on their surface as well. Now, polymer-based nanoparticles effectively carry drugs, proteins, and DNA to target the cells and organs. I mentioned the importance of targeted therapy. So, with polymeric nanoparticles, you can easily target the particular organ or cells. Their nanometer size promotes effective permeation through cell membranes and stability in the bloodstream. So that is another advantage, that the degradation of these nanoparticles is restricted, and that way the drug, the half-life of the drug and also the other degradation effects, that can also be restricted. Now, depending on the method of preparations, we can have polymeric nanospheres and polymeric nanocapsules. Now, what is the difference? Now, polymeric nanospheres, they are the polymer matrix systems where the drug is physically and uniformly dispersed. You see, left hand side, you see these red balls, these are the drug molecules, and you have these polymeric chains forming the polymeric spheres where the drug molecules are physically interacting or physically entrapped within the polymer network. And let me tell you, the polymers which are being used now, used for this kind of research, they are mostly linear polymers, right? They have the linear strand like of structures, linear polymers. So here you can see the linear strands, they are entrapping these drug molecules, and this entrapment is not a chemical interaction, it's only a physical interaction. And we have the nanocapsules where you have a thick polymeric membrane, and within that polymeric membrane you have the drug molecules which are being entrapped within the cavity, right? So in polymer sphere you have a physical network of the polymer, just through physical interaction it is entrapping the drug molecules. And in polymeric your membranes, you have a polymer membrane formed, right? And within that membrane you have the drug molecules encapsulated, right? Now, what are the advantages? These polymeric nanospheres and nanocapsules, it increases the stability of any volatile agents, means if the drug molecules are volatile in cases, and can be easily and cheaply fabricated in large quantities by a multi methods. I will mention that how this can be prepared. There are so many methods by which these polymeric nanospheres and nanocapsules can be prepared. Now, these are significant advantages over traditional oral and intravenous methods of administration in terms of efficiency and effectiveness, right? Delivers a higher concentration of the pharmaceutical agents, that is the drug. The choice of polymer and the ability to modify drug released from polymeric nanoparticles have made them ideal candidates for cancer therapy, vaccines, and contraceptives. Now, it can give you a very effective targeted delivery of the drugs to the specific organs. That is what we call it as a site-specific delivery. And polymeric nanoparticles can be easily incorporated into other activities related to drug delivery, such as tissue engineering. And that is a different area, what we call it as a regenerative medicine, whereby the tissues can be regenerated on polymeric scaffolds, right? Suppose this kind of therapy is again in the research stage, whereby suppose when you have a very fine crack develop in your say bones, so what happens, that crack will not heal by its own. Right now, if you put some polymeric scaffold over there, then that scaffold will accelerate the generation of your bone cells and tissues, and that way the crack will be healed. So that what we call it as a regenerative medication using polymeric scaffolds. So that is another part that is not directly related to the drug delivery, but what we call it as a tissue engineering or what or regenerative medicine. Now, these polymeric nanoparticles, this route also has some, or this technique also has some disadvantage, and these are, first of all, these are costly, because when a new technique comes, of course that is costly, right? That is, when a new way of surgery comes, then that surgical process is costly. Now when more and more people practices, doctors practices, and that is being used in more often, then the cost reduces with time. So that way, when this technology will be introduced, it is going to be costly. And also with low yield. Low yield means that the good quality nanoparticles, good quality drug encapsulated nanospheres or nanocapsules, the yield is low, right? The yield is low. So nowadays, further researchers are working on to develop more quality nanoparticles, that is getting of higher yield of these quality nanoparticles. And productivity is low, as I mentioned. Technology transfer is also going to be difficult. Means when you go for, say, bulk production in industry, that is going to be difficult, because this technology, although it looks simple, but when you will be applying it for bulk production, industrial production, it may not be so simple. Just I can give you one example. This regarding this COVID vaccine, you see that the microbiologists, they develop the vaccine. Some chemistry people may be there. So microbiologists, chemists, they develop this vaccine in the laboratory, but that is not commercially produced over there. Suppose this your AstraZeneca and your Oxford, they develop this Covishield, right? That is a lab work they have done, but the production, mass production, is being outsourced to our that company here in India, right? In that company in Pune, I forgot the name, which is producing the vaccine in India, right? So they are producing in mass. So what means that technology you adopt in the laboratory, you need to further modify that technology when you go for this mass production, and that engineering part is, or that engineering skill is required, and you require the biotechnology people to deal with it, not the microbiologists, neither the chemistry people, right? So that way the technology is transfer for this polymeric nanoparticles, so that technology transfer is going to be difficult, and you require the skilled people for that. So that is the disadvantage. Now what are the polymers that are being used, in the, of course, in the research stage? What are the polymers that are being used? You have the natural hydrophilic polymers, you have the synthetic hydrophobic polymers, because you have both your hydrophilic drugs and hydrophobic drugs. So depending on the nature of the drug, you need to select your polymer. So natural hydrophilic polymers for proteins and polysaccharides. And for synthetic hydrophobic polymers, you have the pre-polymerized materials, and you have polymerized during the process. That kind of polymers you can use for the synthetic hydrophobic drugs. Now, example for proteins, you can have gelatin. Gelatin nowadays are also used in the capsules. The coat is being made of gelatin, but that won't give you a sustained release, right? So gelatin has to be formulated in a different way to make it a sustained or controlled release. So gelatin, albumin, lectin, legumin, and vicillin, these are the examples of proteins, protein polymers. Then for polysaccharides, you can have alginate, dextran, chitosan, agarose, pulanan, right? So these are the polymers, polysaccharides, which are being used. For pre-polymerized polymers which are being used, that is polycaprolactone, polylactic acid, polylactide. It's a copolymer of lactide and glycolide, PLG, and polystyrene. And you can have polymerized during the process. You can have polyisobutyl cyanoacrylates, PBCA, PHCA, polymethyl methacrylates, or PMMA. So these are the polymers where you can start from the monomer and you can polymerize to develop these kind of nanospheres. Now, These are the basic way how these polymeric nanoparticles are prepared. One is the amphiphilic macromolecular cross-linking technique. Another is the polymerization based methods, and another one is the polymer precipitation methods. These are the basic ways. I will not go into detail, just I will mention that way I have prepared the literature over here. In amphiphilic macromolecular cross-linking technique, you can go for heat cross-linking and you can go for chemical cross-linking. Now, what is cross-linking? Suppose when you have the linear strands of these polymeric molecules, you can have cross reaction between the polymeric strands. So that gives you a network. Suppose the fishnet kind of structure. ফিশনেট কাইন্ড অফ স্ট্রাকচার, এই মাছের যে ধরা হয় যে জালটা ওই রকম স্ট্রাকচার, তো ওটা হচ্ছে ক্রসলিং স্ট্রাকচার, ওখানে মাছগুলো বেরিয়ে আসে না ওখান থেকে, রাইট? তো ওই রকম ওইভাবে ড্রাগ মলিকুলস গুলোকে ঢুকিয়ে দেওয়া, সো দ্যাট কাইন্ড অফ আ স্ট্রাকচার, সো দ্যাট ইজ আ ক্রসলিং স্ট্রাকচার। নাও দ্যাট ক্রসলিঙ্কিং ক্যান বি ডান either through application, I mean either application of heat or through applying any chemicals, right? Now for the polymerization based methods, means where you have to polymerize starting from monomers. Now that cross-linking, that previous one, you can do with the already obtained polymers, that is the proteins or polysaccharides, because these are obtained in the form of polymer itself. So you can just apply the cross-linking technique to develop the capsules, nanocapsules and nanospheres. But when you start from the monomers, so you need to polymerize it, right? So for the polymerization based methods, you can have polymerization of monomers through emulsion polymerization or interfacial complexation or even condensation polymerization or precipitative polymerizations. So these are the different ways by which they can be polymerized starting from the monomers. And also precipitation methods, where from the once you develop the polymers, then you go for precipitation to separate and purify the nanospheres and nanocapsules. So the ways are the solvent extraction or solvent evaporation, solvent displacement method, or salting out methods, right? So these are the ways by which you can go for this heat or chemical cross-linking. I'm not going into the detail. I'm just I have given you in points that later on, if you read it, you will be able to understand. So this is your solvent extraction method. It's a single emulsion technique which is being used. You can go for double emulsion technique also, solvent extraction technique. Then solvent displacement method, this is the way it is being done. Salting out technique, by which the nanospheres you can produce. This is the emulsion polymerization, where with the use of emulsifier you develop micelles, and the polymerization takes place within the micelles. And finally, these are the micelles, you see monomer droplets, and the emulsifier, they forms those micelles, and there you have that polymerization going on. And once you polymerize, when the polymerization is complete, then you have to add some salt to break the micelles and precipitate your polymer. So that is the way, and this emulsion polymerization is the most common technique which is being used for polymerizing from monomers, right? But here you need to take care of the chemical initiators, because initiator fragments are likely to stay with the polymer. So you need to use those initiators which should not produce any biotoxicity. So that way you have to be careful with the chemical initiators, right? And this is the precipitative polymerization. So there you need to use some stabilizers, and after polymerization you have to destabilize it. This is the interfacial condensation polymerization, where the polymerization is is taken place at the oil water interface, right? So that is another way of your making the polymers. And these polymer micelles, that what we get in the emulsion polymerization technique, it nowadays it has drawn some special attention, because these micelles nowadays, it develops some special category of polymers for polymeric nanoparticles, which are called polymersomes, right? Polymersomes. Now, what is the speciality of these polymersomes? These polymersomes, they form a solid, not exactly solid, a some kind of a thick and dense outer membrane, and inside it is hollow, right? Inside hollow and a solid outer membrane. This kind of polymerisome structure it can form, and the hollow core, it is capable of absorbing the drug molecules, right? So that way nowadays these polymeric micelles forming the polymersomes, this has attracted a lot of attention, and these are being produced from specialty polymers which is called block copolymers. Now the specialty in the structure that these are the linear polymers, but these has one part hydrophilic, another part hydrophobic. So one part forms the core, another part forms the shell. So that is the beauty of this polymersomes. And because of the hollowness of the core, because it has a very thin core, so most of the part is hollow, so because of that, it is capable of absorbing lot of drug molecules inside, right? Lot of drug loading can be done in the polymeric micelles in the form of polymersomes. And this is a schematic by which the polymersomes are formed. These are being labeled and these are used for the therapy, targeted therapy. Now, the advantages, it gives you these polymeric nanoparticles, including these polymersomes. They gives you better thermodynamic stability in physiological your solution. It gives you a stable and prolonged systematic circulation time within the body, and it gives you a rapid in vivo dissociation. I mean, rapid prevention of the in vivo dissociation. In vivo means within the body, within the cell. That what we call it as a in vivo. And there is another way of experimenting, what is called in vitro, right? In vitro. In vitro means outside the body, outside the living organism, when it is being experimented. Suppose when we develop something, first for bioapplication, first we go for in vitro test, right? Means not within the body, outside, then we go for in vivo, say in the mouse. So when we test in the mouse, then it is in vivo test. And when we are not testing in the mouse, that by but outside the mouse using some certain technology, then it is called the in vitro test. So first there should be in vitro test and then there will be in vivo test, right? So in vivo dissociation of this drug molecules, that is what we call it as the degradation of the drug by the body proteins or enzymes or any other biochemicals, that can be prevented. Now, once you develop these polymeric nanoparticles, you have to consider the pharmaceutical aspects of these polymeric nanoparticles. Otherwise, you will not be able to use them, right? And what are those? You need to purify them because you are using so many chemicals. So you need to purify them, right? So first is the purification, that it should not contain any toxic impurity. You have to ensure that. You have to go for freeze drying of the nanoparticles, right? Freeze drying whereby absolutely dry powders are formed, right? Then sterilization of the nanoparticles. You will have to sterilize it before you load the drug in it, right? So these are the pharmaceutical aspects you need to consider before you go for this, application, application study. Now, once you have this, once you ensure these pharmaceutical aspects, then you can go for now the characterization of the polymeric nanoparticles. That is a parallel study you need to do before you go for application or absorption of the drug molecules within it. So you need to characterize them for particle size and particle size distribution, surface area, surface chemistry, hydrophilicity and surface charge density, purity and quality, stability, that is on shelf and upon administration. Shelf means how long you can store, right? That what we call it as a shelf life, right? How long you can store. So the shelf life is given, that is the, suppose for the drug it is written as the expiry date, so that is the shelf life, how long you can store. And upon administration, how long it is stable within the body. Then percent entrapment efficiency, entrapment of the drug molecules, how much drug it can entrap, right? So that you need to find out entrapment efficiency. And the yield, polymeric nanoparticle yield, what is the yield of the nanoparticle, that also you need to consider. And in vitro and in vivo releases, first in vitro, then within the body in vivo releases, this study. Now you can this size evaluation, for the size evaluation you can go for TEM study, you can go for ACM study, and you can go for dynamic light scattering study. Now, when you go for dynamic light scattering study, you can also determine the zeta potential or the surface charge. And for the spherical shape, exact spherical shape, you can study from your atomic force microscopy, particularly for nanocapsules, right? So these are the ways you can study, and TEM can give you the picture of that polymersomes. If you have formed polymersomes, whether the polymersomes are formed or not, that core shell structure you can see under transmission electron microscopy, and it should be a HRTM, high resolution transmission electron microscope. There you will be able to see it, right? Now for surface charge, as I mentioned, you can just measure it from the DLS study. Then for regarding the molecular weight, you have to go for GPC, gel permeation chromatography. For structure determination, structure means that crystallinity, that also gives you a morphology study. Then you can go for dynamic your sorry differential scanning calorimetry, DSC, or differential thermal analyzer, DTA, TGA, thermogravimetric analysis, that can give you the thermal stability of the polymeric nanoparticles, and wide angle X-ray diffraction study regarding the crystallinity. Crystallinity study you can do both from DSC and waxed. Okay. Now these are the way by you can calculate the drug entrapment, that is the mass of the drug entrapped within the nanoparticles by mass of the drug used in the formulation. That is the drug entrapment efficiency. Now the mass of the drug you can find out. Actually not the mass, we actually calculate in terms of concentration, and the drug concentration, how much it is entrapped with time, and when there is a saturation in entrapment, we can find out by the UV absorption study of the drug molecules, right? We take a saturated solution of the drug and we put the nanocapsules within it, or nanospheres within it, and from the saturated solution we just measure the peak intensity, UV peak intensity of the drug molecules with time. Now gradually it will decrease, right? Because it will be absorbed within the nanospheres or your nanocapsules. So within the solution, the intensity of the drug peak, that absorption peak lambda max, will decrease. So when it gets stabilized, so there that point we will consider that the nanospheres or the nanoparticles, they have absorbed maximum amount of drug molecules. So that way the efficiency is calculated in terms of concentration, and this is the calculation of the polymeric nanoparticle yield after absorption of the drug, that is actual weight of the product and total weight of the excipient, that is the nanoparticle and the drug. So these are the calculations and characterizations we need to do, and this is the in vitro study. For drug release, we have a cell, what we call it as a France diffusion cell, where we go for this in vitro study, that is outside the body study, where your cellulose acetate membrane, what we call it as a cellophane membrane, is used as a barrier, and the nanoparticles are entrapped within it, and the concentration of the drug released from the nanoparticles through these cellophane membrane within the solution, right? That solution temperature is kept at 37 degrees Celsius, which is the body temperature, and then a buffer is used, right? So that way the how much drug is coming in that solution with time, it is being measured, and again it is measured in that UV absorption mode, the lambda max. So that way the concentration determined with respect to a calibration curve for a particular drug, and that way we can find how much of the drug is being released with time from the nanospheres or nanocapsules. So that is the in vitro study. And for in vivo study, we need to further go for this kind of evaluation. First of all, the plasma protein absorption and the liver spleen RES study. Now RES is the reticuloendothelium system study, and this is a speciality of the liver, that whenever some foreign particles is administered within the body, these group of cells which form this reticuloendothelium system, they will try to eliminate that foreign body from the from your body, right, through the kidneys and all, right? So you need to, your body should identify that these polymeric nanoparticles, these are not foreign bodies, right? So these are, I mean, required for your body. So the RES system should not become active to eliminate this from the body immediately. So it need to, I mean, those cells need to recognize that this is not a foreign body and this is not going to disturb you, right? So that identification of the polymeric nanoparticles to your body, that should be that should be important. So for any polymeric nanoparticle, if RES activity is high, then you cannot use it for drug administration. So that you need to pass. That is the first thing. Then you need to study the phagocytosis by the cells, right? Phagocytosis means when there is endocytosis, that is the cell is taking up that polymeric nanoparticles containing the drug, right? And how long it will allow it to remain within the cells and allow the drug to release. Now if the cell does not allow that nanoparticle to stay or remain within the I mean cells for long and release it, and that release process is known as phagocytosis. So if the phagocytosis is very fast, then after going inside the cell, the nanoparticles will not get enough time to release the drug. So that is the second point that need to be cleared. So these are the two major points. So for that, these polymeric nanoparticles are to be surface engineered with some agents which the body identifies, through which the body identifies that no, these are not harmful to us and the cells will not exude it very fast. So that way the polymeric nanoparticles are engineered, or what we call it as a labeled, through different materials, like say antibody coating, some liposomal coating. So these are the molecules which are further attached with the polymeric nanoparticles to make them recognizable by the body. So that what we call it as a your surface engineering. Now along with this surface engineering technology, we can further go for, or we can further enhance the targeted delivery mode of this nanoparticles. That is, for the particular organ where the drug is to be administered, we can increase that efficiency. And if we reach specifically to that organ, your nanoparticles, if it can reach specifically to that organ, then almost all the drug molecules are being carried to that organ, and the efficiency of therapy will be enhanced. Very to a very higher level. So suppose these antibiotics nowadays, suppose we say that you have to take it for 5 days, these antibiotics. So if you can ensure this kind of site specific delivery, maybe within 2 days you will be cured, and the antibiotic therapy will then be reduced to two days. You don't have to require for 5 days, right? So that way the therapeutic efficiency can be improved with this targeted delivery. Now once you go for this targeted delivery, which is also a part of what we call it as a nanomedicine, because this is being achieved by the use of these nanospheres and these nanocapsules. So these what we call it as a part of nanomedicine. So here it deals with the interactions of nanomaterials, that is the surface of the particles, or analytical nano devices with living human materials in this technology, right? And it is an extremely large field ranging from in vivo and in vitro diagnostics to therapy, including targeted delivery and regenerative medicines. Now, this technology, this targeting, we also call it as a clever mode of drug delivery, because it gives a coordination of these three functions. What are these? First one is the recognition and binding to the target. Second one, the drug carrying of the drug. And third one is the therapeutic action at the specific site by release of the drug. Right. Now what targeting moieties can be used? You can use antibody attached with the polymeric nanoparticle. You can use specific proteins attached with the polymeric nanoparticles, or lipoproteins, or any other biological substance. Suppose let us take the example of protein. Suppose you need, if I give you the example of this cancer therapy which are being thinking to be implemented in future. So that targeted, I mean, tumor cells and all. So these tumor cells, they have a particular pH around ambience. That ambience has a different pH from the normal cell, as well as these cells will synthesize a different kind of proteins, right, from the normal cells. So if you label your polymeric nanoparticles with some protein which can identify those proteins which are being secreted by those your cancer cells, then what happens, these polymeric nanoparticles will be drawn specifically to those cells, right, with the drugs. So that way the targeting is being achieved, right? So attachment of protein with this polymeric nanoparticle or any other biochemicals with polymeric nanoparticle helps them to identify those cells, because the signals those cells signal is what? Signal is nothing but the secretion of some specific proteins by the specific cells, right? So if the your surface molecules of the polymeric nanoparticles or the labeling molecules of the polymeric nanoparticles, they are containing the drugs, if they can identify those signals, then they will be drawn to those sites and the drug will be released over there specifically. So here the steps is that interaction of the nanostructures with plasma proteins, then adsorption of nanostructures to cells, right? Then uptake and recycling, that is trans endocytosis and endosomal escape of the nanostructures from the cells. Then your safety evaluation, that is your in vitro in vivo study, that should be complete before application, and in vivo biodistribution study, that is whether it is released rapidly by the RES system or not. So these two studies is essential, and first two is the method by which these nanoparticles targeted therapy they acts, right? Now these kind of nanoparticles targeted delivery, they exhibit a higher intracellular uptake. Means if you target them for a particular cell, then the cell will happily allow these nanoparticles to enter within them, right? So these intracellular uptake is higher, right? Now suppose it may so happen that it is being targeted, but it does not reach the target, or the cellular uptake is not high, then actually the therapy will not complete. So these are the different steps where you need a coordination between each and every step to get a very successful therapy. So the technology is not simple. The step by step you have to achieve the success. So in this targeted, when the polymeric nanoparticles are engineered with these targeting molecules, then they can penetrate the submucosal layers, while the microcarriers are predominantly localized on the epithelial lining. So if you do not target, or if you use a microcarriers, any sort of non-polymeric microcarrier, then they will lie on the surface of the epithelial tissues. They will not penetrate the cells like these nanoparticles. So here it can also administered into systematic circulations as well, means the blood. So you can also go for the systematic circulation, means just you can inject in the blood, right? And after injecting, once it moves into the blood, then it can be easily carried without any degradation to the targeted cells. So these are the advantages you can get, and you see how this is a polymer and how it is linked with this kind of a targeting molecule. So these are the ways it is being synthesized, and you see that once it is being targeted, then it carries the your drug molecules to the specific cells, and it is being released. And this is one way of making these nanoparticles targeting, what we call it as a pegylation, that is using polyethylene glycol. Polyethylene glycol is a very good molecule for, which is being used over these nanoparticles for further attachment with the targeting molecules, right? And you see that the half-life for a drug, while you make this kind of a polymeric nanospheres and nanocapsules, from 30 minutes to it can be enhanced to 5 to 10 hours. So you see that the 50% concentration, when it was reduced by 30 minutes, when it is administered using, say, gelatin capsule or something like that, But when it is being put in a nanocapsule, polymeric nanocapsule or nanosphere, then the half-life is increased to some 10 hours, from 30 minutes to minimum 5 hours. It is enhanced. So 4.5 hours enhancement in half-life minimum we get, right? So these sort of advantage. So this is, you see schematically how it is being targeted, and this sphere, this is the nanosphere, followed by the PEG molecules and then the targeting molecules, and this is the overall entity that it is being synthesized and it is being tried to deliver to the on-site. Okay. Mode of action, just what we have mentioned, that is need to identify the receptors, and then only it will be targeted, and that way the surface modification of the polymeric nanoparticles are being done. And this is a table that shows the what are the polymers that are being used to form these nanospheres and nanocapsules for targeting, right? And in different therapy, suppose in cancer therapy you need to deliver the anti-cancer drug, anti-cancer agent, then intracellular targeting can be done, vaccine adjuvant, right? How to form the vaccine adjuvant, so what kind of polymers are being used, DNA delivery in gene therapy, what we call it as a gene therapy, to repair the defective gene, right? Gene therapy, ocular delivery. So this shows you that how it is being done nowadays, means in the research stage, of course. Now, the challenges in this technique is the prevention of drug from biological degradation, effective targeting, patient compliance, cost effectiveness, and product life extension. Now, I'm coming to the last part. Just only say 5 minutes, I will take more. I will talk about a different kind of polymers which can itself act as a drug, and these are the work that we have done in our laboratory. And this is what we call it as an architectural polymer, what we call it as a hyperbranch polymer. Now these polymers are not linear polymer, right? These polymers are not linear polymer. It has this kind of shape. You see, there's dendrimer. Dendrimer is a very symmetrical hyperbranch polymer, whereas hyperbranch polymer is unsymmetrical. So when you have these kind of polymers, which is not linear, but which has many branches, almost it is a branched polymer, fully branched polymer, but unsymmetrical, then what happens? For every branch end, you can develop the functionality. So when you develop the functionality, and these kind of polymers with the branches, it has many holes inside where you can entrap, even if you want, you can entrap the drug molecules, or this polymer itself can function as a drug for specific cases. So we have prepared this kind of polymers in our laboratory, and we use them as these drug molecules. Now in 2017, we published this book from Springer. In fact, they agreed to publish, whereby we have shown the efficacy of these hyperbranch polymers as biomedical agents, right? And here we have discussed thoroughly that how these polymers are synthesized and what latest applications it is potent of, right, for in future. Now I can directly go for the results that I want to show you. So we have actually used a very simple kind of your monomers to start with. We have say used polyvinyl alcohol and citric acid. You know that citric acid, citrates, these are very good antimicrobial agents, means it can act on the microbes to kill them. So we have started with this citric acid and polyvinyl alcohol, and we have formed this kind of hyperbranch structure, and we have characterized them, this hyperbranch structure, using NMR, IR, viscosity and all, and we established the hyperbranch structure, right? And then we applied it as a therapeutic agent, as a drug release agent, right? As a drug release agent. So this is the first application where we have entrapped the drug molecules within it, and we have studied the release of the drug molecules in vitro, not within the body, that in the France diffusion cell. So this is where we entrap the drug within it, and we see the release at a constant rate. This blue one, that is the lowest one where we have used the most hyperbond structure for encapsulation of the drug molecule. So there we see that the release rate is the slowest. So you can get a more sustained kind of a therapeutic effect while you apply this kind of a polymer. Now, this is the another system that I was mentioning, where we have used citric acid and polyethylene glycol to form this hyperbond polymer. So we have developed this kind of structure, and these polymer we have used directly as a therapeutic agent. So this is the characterization of the structure. Is a IR NMR by which we establish the structure, and this is the particle size determination using your dynamic light scattering. And here you see that the polyethylene glycol having a molecular weight of 4000, it gives you a average size of 4000 nanometer, whereas when you are polymerizing it, polyethylene glycol with citric acid, and when you are forming that hyperbond structure, then the size reduces to only 13 nanometer, 13. So you can imagine a drastic change in structure, right, and size of the polymer when you are making it in the branched mode. And here, this is the rheological study, and here you see the antimicrobial effect. So these are the bacteria killing effect. We have studied with both your gram positive and gram negative bacteria. Gram negative bacteria is E. coli, and gram positive bacteria is the B. subtilis. So with both these bacteria, we have seen our polymer is very effective in killing them. So this is the in vitro study, but later on we have done the in vivo study in mice also, and we have seen the very strong effect of killing the infection or reducing the intensity of the infection with this polymer itself. So here that polymer itself acts as a drug for therapy, right? So these are the further study, that what we call it as a cell viability study. And what that cell viability study is, that when you are applying it within the body of the mice, then whether these polymers are killing the healthy cells or not, right? So that efficacy need to be studied, and we have seen that almost it is 100%, means it is not killing the healthy cells, rather it is killing the cells which are diseased, right? So that way we established that our polymers can be safely used for in vivo applications. So these are the further antibacterial efficacy study. So that way we established that this polymer can act as a nanomedicine. So what are the excerpts of today's discussion? We have come to the end of our talk, that is sustained drug delivery using these polymeric nanoparticles is the need of the hour, basically, to make the conventional drug administration to be more effective. Targeted drug delivery using polymeric nanoparticles to increase the efficacy of the drug molecules. And the last part, that is exploration of the differently structured polymer in forming these nanoparticles, can further increase the scope of using these polymeric nanoparticles directly as a therapeutic agent. So with this, I finish my talk, and thank you very much to all of you for your patience hearing. Now, if you have any questions, you can ask me.
Thank you, sir, for your illuminating talk. I am sure all of us have been enriched with the knowledge of this faculty improvement program. Participants may feel free to add their questions in chat box or may ask.
Any question, any comment, any opinion, you can share, no problem.
I have a question.
Yes,
please.
I've heard of liposome encapsulated drug delivery.
Yes,
Does
yes.
it fall in the same category as polymer nanoparticle?
Yes, see, liposome basically is used as your targeting moiety. Yes. Right, targeting moiety means you have to develop that polymeric nanoparticles that you need to develop, and as I mentioned, that you need to target it. Once you suppose if you do not want to target it, just if you go for sustained delivery, then you don't have to use liposome. right? So liposome only for targeting it to certain organs, right? Or it is liposome, what we call it as a signaling or signal, identifying molecule. Liposome is used for that purpose.
Another question is that after the drug is delivered, what happens to the vehicle? Like there should be a lot of, like
Yeah. See, it will be excreted. It will be excreted, right? So that is another part we need to consider. That is the excretion, because the cell will release it there because of the phagocytosis, right? Because of the phagocytosis mechanism, it will release into the blood, right? And you need to ensure that it goes out through the excretory route from the body. So that is also considered. And for these, polymers that I have mentioned, for all of them, the excretion is not an issue. That part is tested.
I have a question, and thank you very much for your such a nice presentation. So my question is that the polymer, all these polymers may have some toxicity.
No, polymers are not toxic. Let me tell you, polymers are not toxic unless it is made in the nanoparticle mode. It is not being taken up by the cells, right? So this is you see, means it's a wrong perception. Polymers are toxic. No, if it is in the microform, it will not be absorbed by the cell. So that way it cannot develop the toxicity. Now the toxicity, see that part has to be ensured. No, as I mentioned that first of all you need to ensure all those in the in vitro form, that whether it is toxic, whether it is hemolytic, whether it is killing the healthy cells or not. So all these in vitro studies first you need to consider, then only you go for in vivo.
No, my question is that if the polymer have some some amphiphilic parts, so it can interact with proteins, because protein always accept some amphiphilic molecules. So there may be some toxicity because these proteins are interact with these polymers.
No. Whenever protein interacts with the amphiphilic molecule, it cannot, means it not always it generates toxicity.
Obviously.
No. See, for these you have to ensure that the biochemicals are not producing any toxic effect. You have to ensure, in cases it may, but not always. That is my point. Not always. And you have to use those kind of polymeric nanoparticles, right? So these are the primary tests before you apply these for this kind of applications. That is the primary test you need to do.
And the second question is, what is the stability of these liposomes or this sort of polymers in the plasma?
Stability.
Yes.
That stability. See, you have to ensure the stability until that point when it is being carried to the target point. So you need to calculate that, and you have to use that kind of attachment. Means if the attachment is only physical adsorption, it may not be stable to that point when it is reaching the target organs, right? So there you might go for some sort of a much stronger secondary interactions. You cannot go for a primary interaction, then it will not detach, right? So you have to go for some stronger secondary interactions. So that way you need to ensure, but the stability study is being done, that is from till it is being carried to the target organ. So you have to see that whether your nanoparticles with that liposome is being stable until that time or not. You have to study it. See, at this moment, I cannot see it is stable for 2 hours or 3 hours, right? But I can tell you how the stability study is being done. That I can tell you.
Okay, and my another question is what is the capacity of this polymer to deliver a drug, or how much drug it can contain inside it, and is there any process to measure it?
Of course, drug entrapment efficiency. I have said that that drug entrapment efficiency, that you need to measure at first, no, whenever you develop a polymeric nanoparticles, and when you ensure that it is not toxic, it is not hemolytic, it will not interact with the proteins producing any toxic effects and other, interact with proteins and other biochemicals, then you go for that entrapment study. So for that entrapment study, what we do, we take a saturated drug solution, right? And you have first, that you need to find what is the lambda max of that drug molecule. Now you put your polymeric nanoparticles, nanospheres within that saturated solution. Now what happens? It will gradually first adsorb, then absorb those drug molecules, and in the solution the drug concentration will decrease. Now the decreasing concentration you need to find out from that reduction in the peak intensity from UV. So that way you need to correlate, right? And if you have a calibration plot, then you can find out that how much drug was encapsulated. So that way the entrapment efficiency can be calculated, and that entrapment efficiency varies. Suppose if we use a nanocapsule and if you use say a nanosphere, what happens in a nanosphere? Only your vacant points within the linear polymers, these are available for entrapment of the drug molecules. Whereas in nanocapsule, you have the inner core which is available for the drug entrapment. So generally, nanocapsules have a higher entrapment efficiency than the nanospheres. Now, if you go for that polymersome I mentioned, micelles, so they have a, hollow, complete, almost a hollow core. So there the entrapment efficiency is further. So a polymer micelle carries more drug compared to a nanocapsule, and again nanocapsule carries more drug compared to your nanosphere.
Okay, and it is my final question.
No problem. You can carry on. No problem.
Okay. The question is, if I see you use one citric acid molecule in your polymer.
Yes.
So what effect?
Yes.
The stereochemistry of the citric acid in the structure of nanoparticle, nanoform of the dendrimer.
See, we at this moment we are not concerned with the stereochemistry of citric acid, because first of all we need to find out whether it is effective as a drug or not.
Okay.
Right. So that's the primary study we have done. And the idea behind using citric acid was its antimicrobial effect, right? Because citrates, these are used as a natural preservatives, right? Yes. So from that idea, we use citric acid. Now later on, to find out the efficacy, when we will be studying on the efficacy part, so there we will be considering the your stereochemistry part. So, but at this moment, we have not considered.
Okay, thank you very much. You're welcome. Such a good presentation and very well, and it is very informative for us. Thank you very much.
Thank you. Welcome.
Okay, good evening, sir. I have
Good
two
evening.
questions.
Please.
One is related to what Naeem was asking, that did you use a copolymer of citric acid and polyethylene glycol? What did you use?
Yeah.
It's
It's actually not a copolymer. It's a condensation polymer. See, condensation polymers are not copolymers. For copolymers, we have a different theory, right? These are condensation polymers, because citric acid cannot alone polymerize. You need an alcohol, OH, to polymerize. So it's a natural way of polymerization of citric acid or any acid through condensation reaction. So it is not a copolymerization. It's simply condensation polymerization.
My question is, why did you choose citric acid and polyethylene glycol? What's the reason behind your choice, other than the fact that citric acid is a known antibacterial or some such properties? Why didn't you choose any other acid or any other alcohol? Just because of polymerization property of polyethylene glycol?
No. Polyethylene, I have not mentioned. Polyethylene glycol also has a very strong oxidizing effect. It's a very strong, it's a very strong, what we call it as a Ross generating or Ross instigating polymer. Ross instigating effect it shows. Ross means your oxygenated species, reacting oxygenated species or radicals.
That means what should I say? Free radical. Free radical.
Free radical that can kill the cells, right? That can kill the cells,
That's
right? So polyethylene glycol has that effect, right? So that was the reason that we have combined polyethylene glycol with citric acid. Otherwise, we could have used glycerol, right? Glycerol we could have used, or ethylene glycol we could have used, but we have, specifically taken polyethylene glycol because of this Ross generation effect.
property. Okay, thank you. My second question is, you have said that control release is used by using, is control release of drug increased by using polymer nanoparticle?
Yes, of course.
Then
Of
why?
course.
Why is it so? Why does
See,
surface area increase?
when you are using in this current formulation, suppose these tablets, right?
Okay.
How these tablets are being made? You take the drug molecules and put some binder, then compress it, and the tablet is made.
Okay.
Right. When you prepare these capsules, right? How do you make? You entrap these capsules within a gelatin coating, and once you put it, that gelatin is water soluble. Immediately it will melt, and the drug molecules will come out. Right. So this is a very fast release of the drug molecules within your plasma. So its release is not controlled. It is open, almost it is open. But once you cover it with some polymer, a coating of polymer, say, right? Once you cover it, then because of the diffusion effect, then the release is controlled.
Because of diffusion effect, the release is controlled. Okay, and it just because for nano dimension particles, just the increase in surface area leads to this control.
Yes.
Improved property of diffusion.
Not really. See, nano. When you suppose if you go for a microparticle, right? The microparticle. It's. What you can say that when you administer in the form of microparticles, then it moving into the your plasma, then carrying to the cells. In fact, there is a fixed dimension for the cell membrane to allow anything to move inside, and there is a restriction of dimension 250 to 300 nanometer. If anything you supply more than that size, then the cells will not allow for endocytosis. Right. Okay. So that is another reason for using nanoparticles. And also for microparticles, what happens? The surface area will not get much higher. So the drug loading point of view, and also the cellular uptake. So these are the points for which we go for polymeric nanoparticles or nano dimension.
Okay. Thank you so much. Thank you so very much for such a nice presentation. Thank you.
Welcome.
Okay, can I ask another question?
Please.
This as you mentioned, the idea of polymer nanoparticle being applied in drug delivery is still at its research stage. Did I get it right?
Yes.
So right now, at the present point of time, the sustained release drugs that are there in the market, what technology is it based on? It's polymer nanoparticle technology, is it?
So the coating which is being used, that is being made, a bit thicker, actually, so that the dissolution time becomes higher. That coating
manipulating the solubility of
only that.
And the half-life period is also increasing because
not really. Suppose these doxed, when there was during the first stage of COVID, the dox was prescribed 2 doses per day.
Yes.
Right. So not really. Maybe it is improved. See, whenever the dissolution time is increased, the half-life period will certainly increase, but not to that extent that we have discussed here by using polymeric nanoparticles.
Okay, understood.
Dr. Bandyopadhyay,
please.
I am Mustafizur Rahman, faculty member of this department. I have not a question, just a sharing. In your first part of talking, you talked mostly about drug doses. I think nowadays doctors are not aware of such drug dose. they often go for trial and error. My
কেন বলুন
feeling is
তো
like this.
কেন বলুন তো আমি আপনাকে বলছি ডাক্তাররা তো দে আর নট অ্যাসোসিয়েটেড উইথ রিসার্চ ডেটা
Yes, yes,
তারা কেউ জার্নাল মেডিকেল জার্নাল তারা পড়েন না তারা শুধুমাত্র মেডিকেল রিপ্রেজেন্টেটিভদের সঙ্গে অ্যাসোসিয়েটেড they are only associated with medical representatives for selling of the basically ওরা ডিস্ট্রিবিউটর এরা হচ্ছে এজেন্ট ডক্টরস আর nowadays they are agent they are not
আমার একটা সন্দেহ জাগে তাদেরকে এটা শেখানো হয় না ওদের কোর্সে
শেখানো হয় কিন্তু তারা টাকা রোজগারের তাগিদে সব ভুলে যায় ঠিক আছে না মানে যেটা সত্যি কথা আমার বলতে আপত্তি নেই মাল্টিপারপাস ড্রাগ কত অ্যাভেলেবেল একটা ড্রাগে আপনার কতগুলো থেরাপি হতে পারে সেগুলো কিন্তু সব আজকে আমরা লিটারেচার পড়ে জানতে পারছি আর ডাক্তাররা জানে না এটা হতে পারে ডু ইউ বিলিভ দ্যাট
এই থ্যাংক ইউ ভেরি গুড থ্যাংক ইউ ডক্টর বন্দ্যোপাধ্যায় থ্যাংক ইউ
I have one silly question, sir.
Not
there
silly,
any
please.
chance of long-term adverse effects of these COVID vaccines in human body in near future?
দেখুন এইটা সম্পর্কে আমার তো রিসার্চ অভিজ্ঞতা নেই আমি যতটুকু লিটারেচার পড়েছি হোয়াট এভার লিটল আই হ্যাভ রেড দে সেড দ্যাট দে হ্যাভ used some biomimicking molecules and it is prescribed that even pregnant women, persons with comorbidity, সবাইকে এটা অ্যাডমিনিস্টার করা যায় সেটা ওরা বলেছে এবং সেটা করাও হয়েছে কিন্তু লং টার্ম কারণ আপনার অ্যান্টিবডি স্টেবিলিটি তো থাকছে 6 মাস ঠিক আছে যে অ্যান্টিবডিটা তৈরি হচ্ছে
কোনো কিছু হতে পারে তাই তো তার
হ্যাঁ ইনিশিয়াল ফেজে একটা দেখুন একটা হচ্ছে যে এই ভ্যাকসিনেশনের পরে whenever there is a vaccination you might feel a little fever so that is natural that when some foreign body is injected within your body the body will react obviously it will react so it is that reaction effect but the antibody which is being developed after 15 to 20 days of administration of the vaccine that is being said to stable up to say 6 months after 6 months there is nothing left right so that is why in America এখন তো আপনার 4th বুস্টার চলছে
হ্যাঁ
আমাদের এখানে তো এই দুটো দিয়েই ছেড়ে দিয়েছে ঠিক আছে 4th বুস্টার চলছে
আচ্ছা why this in third booster this dose is half dose
না আপনার প্রথম বুস্টারে প্রথম যখন টোটাল না 14 ml ওরা অ্যাডমিনিস্টার করেছে দুবারে first in your first vaccination they have administered 7 ml and in the second dose 7 ml
yes
right so total 14 ml they have administered twice
okay হ্যাঁ
7 করে ভাগ করে দিয়েছে
7 করে ভাগ করে দিয়েছে ওকে
এবং এইটা অনেকটা আমাদের এই ট্রেড ব্যাকে যে ভ্যাকসিনটা দেখবেন যে জিজ্ঞেস করে 6 মাসের মধ্যে কোনো ভ্যাকসিন নিয়েছো কিনা মানে কেটে ফেটে গেলে সেপটিক অ্যান্টিসেপটিকে এটাও ঠিক সিমিলার মানে এটার ফেটটাও সেরকম যে 6 মাস হয়ে গেলে এটার আর এক্সিস্টেন্স থাকছে না ট্রেড ব্যাকে যে ভ্যাকসিনটা দেয় সেটা 6 মাসের স্থায়িত্ব তার
okay sir have a thank you have any questions from the participants any more question I think all of us have got answer all of you have answered
I hope I have not bored you I have not bored you because it's not real I mean entirely chemistry polymer is beyond our conventional chemistry so that way I think I have not bored you in this evening
Not at all
not at all not at all not at all
okay now I may request
no it's okay please carry on
now I may request Dr. Mustafizur Rahman our faculty member for vote of thanks
thank you Monisha madam good afternoon to all on behalf of our principal madam Professor Shivli Sarkar faculty members of this department and other departments who are present here our staff members I express my heartfelt gratitude to Professor Abhijit Bandyopadhyay for delivering such an illuminating and encouraging speech tonight. Despite his tight schedule, he agreed to throw some important light over such an illuminating and important topic. We, the faculty members, will definitely be benefited over thinking on such subject in future. I thank our Madam Principal, who always extends her helpful hands over organizing such webinars. We extend our thanks to Department of Biotechnology, New Delhi, for funding this as well as nationwide such programs. I thank Mr. Arvind for providing his sincere technical support in conducting this program. I also thank our head of the department, Dr. Papia Ganguly, who always keeps her positive attitude over such programs. Thanks also to our program coordinator of this program, Dr. Monisha Mukhopadhyay, for thinking over such an interesting and important webinar. Thank you all again. Good night to you. Now it's over to Monisha Mukhopadhyay.
I have a request to all of the participants. Please fill up the feedback forms which has been provided in the chat box. Now this ends our program. Thanks you, sir. Thank you again. Thank you so much.
And good night.
Thank you all.
Good night.
Okay, good night.
Good night.